All right, good evening everyone, and thank you all very much for attending tonight's Research Tuesdays event. This night's event is called Cracking Cancer. My name's Kalia Primer and I'm doing my PhD here at the Adelaide Medical School, and I'm also your MC for tonight. I would like to start by acknowledging the Kaurna people, who are the original custodians of the Adelaide Plains and the land on which the University of Adelaide's campuses at North Terrace, Waite and Roseworthy are built. So tonight's presentation is going to be all about the University of Adelaide's newest centre and research institute, the Adelaide Centre for Epigenetics, or ACE in short, and the South Australian Immunogenomics Cancer Institute, also known as SAiGENCI. So launching mid last year, this centre and the institute are really about bringing the best minds in epigenetic and cancer research all together under one roof. The centre and the institute are going to be home to cutting-edge technology and brilliant researchers from around the globe to deliver knowledge in the exciting epigenetic field and also a bench-to-bedside approach to cancer treatment in South Australia. So during tonight's event, you're going to hear from two of our new recruits, Professor Jose Polo and Associate Professor Luciano Martelotto. They're going to discuss their plans for ACE and SAiGENCI and the expertise that they've both brought to their new roles. And following their presentations, you will have a chance to ask some questions of each of them. We'll be taking questions in person, but also online. So I encourage our viewers, if you're watching online, please do submit your questions via the Q&A section down the bottom of your screen. So let's get underway. I would first like to introduce you to our first speaker, Professor Jose Polo. Jose has two hats. One of these is what he wears as the newly appointed director of the Adelaide Centre for Epigenetics, and the other is as a program lead in the SAiGENCI's Cancer Epigenetics program. Professor Polo's work in epigenetics spans several fields of research, such as nuclear reprogramming and cancer, where he has investigated the role of the epigenome in the cell identity across different fields. And Jose's research has been very widely published, including in prestigious journals such as Nature, Cell, Nature Medicine, Nature Immunology, Cancer Cell, and Nature Neuroscience. These are heavy hitters. So I'm sure we're all very excited to hear what he has to tell us about tonight. Please join me in welcoming Jose to the stage.
Thank you for such a nice introduction. And thanks to the organizer for giving me the opportunity to present our work and tell you what we do. So I will just start with a little bit of developmental biology, and then I will tell you what is this epigenetics thing. So as we know, the oocyte gets fertilized and give rise to the zygote. That continuous development, this is one cell that continues development into the blastocyst, and the blastocyst become, in this case, a trilaminar embryo, and if everything goes well, you get a beautiful baby. Now, if you get really lucky, you get a twin baby that are sitting here, and these are my kids 11 years ago. So now, something that is amazing about them and about all of us is that we are made of hundreds of different cell types. They look different, they have different functions. There you have a neuron, a blood cell, and a kidney cell. And we know that all of them came from one cell. So that means very clearly that information to make all the cells of your body, it was already in this cell. Yes. So a big question was, what happened with that information during development? And many years ago, Sir John Gordon did an experiment where he took the nucleus of one of those cells, an adult cell, and put it back into an enucleated egg. He did this with frogs, and he was able to make a new frog, or to rebuild the frog. And that means that all the information to make an entire organism still present in every one of your cells. So that's amazing, because when we see our cells, if you see now your hands, or blood, or all the things that you see, they all have the same information. So it's not the genes that make them different, because they all have the same genes. And if you are going to take anything out of this talk, please check that all the cells of the body have the same information. So it's not the information what makes them different, and they are very different, is how the information is managed. Yes. By the way, From the blastocyst, inside the blastocyst, that little ball there is called the inner cell mass. This will give rise to all the organism, and this is where the famous embryonic stem cells are derived. And then we go back to that in a second. So what's the I call the gene library. So your body, this is a chromosome, is full of these units of information that we call genes, yes? And these genes, in a way, are like books with different books. And we have around 20,000 of these genes, these books with information exactly to produce a protein that has a function, yes? And as I said, we have thousands of these genes and books that some of them are open and some are closed. And the difference between two cells, again, is not that they have different books, is which books are open or closed, which genes are open or closed. So if you look at an embryonic stem cell and you look at a skin cell, they have exactly the same genes, but in some cases, some are open and the other ones, some are closed. And that's what makes a cell different from the other one. So at the same time that John Gordon was doing that experiment, Conrad Waddington was thinking on this problem, was thinking, how can be that this happens? So he came up with this theory about the epigenetic landscape. Epi means on top of the genes, yes? So he came with this idea where the genes were in this landscape of possibilities of the genes, yes? Which ones were open or closed, let's say. And you have, for example, here a cell of that inner cell mass that gives rise to the entire organism or embryonic stem cell that during development starts to adopt open, close, different genes open, different genes closed. And you end up with all the different mature cells of the body. And if you see how this happens, you have an embryonic stem cell. What happens is, for example, you can differentiate it into, in this case, cardiac cells in the plate. And again, these are the same cells. Yes, they have the same information. It's just that we just change which information is used. Yes, and here is an embryonic stem cell, and when we put it here, it starts beating like a heart cell. So the way that this is, he still thought in, okay, how this landscape could be dominated or produced. And he thought he just hypothesized that should be some lines that were pushing these genes into different positions of open and close, and there have to be something that acts like pegs that anchor those lines to build this different landscape. And of course, this was like 60-something years ago. And now, 60 years later, we know what they are. So these epigenetic changes are chemical modifications that your DNA, yes, and the proteins that surround the DNA acquire in order to open or close the gene. And this is managed, this is driven by a type of proteins that we call transcription factors. And some transcription factors will activate some genes, and some transcription factors will repress it. Basically, they will close it. And these transcription factors, I said, they work like a librarian in a library. So imagine that you go to the School of Medicine, and it has exactly the same library, the same books. The library of the School of Medicine has the same books that the library of the School of Law. Now, in the School of Medicine, the librarians of the School of Medicine will only open for you your medical books. So when you go to study there, you become an MD just because the books that were open are the MD books, the medicine books. When you go to the School of Law, the librarians in the School of Law only opens the law books. So sadly, you become a lawyer if you stay there. Now, as you can imagine, if you can manage those librarians, you can manage the identity of a cell. So if you just go to the School of Engineer and you throw inside a medical librarian, all your engineers will become MDs. So this is what we do. That's what I specialize. We call nuclear reprogramming. We have learned with transcription factors, the librarian control these different programs, different books. And what we do is we manipulate them to create different cell types. And the most famous one is probably the one that you can grab a cell, any mature cell, skin cells usually, they are easy to get, and you can convert it into any other cell type. You can convert it into a blood cell, a cell of your brain, Or you can even convert it into a cell, an embryonic type of cell, yes, like cell, which is called an induced pluripotent stem cell. And basically what you do is you check a skin fibroblasts, you throw 4 transcription factors in this case, they work, and they just open and close the different genes, and these genes change, and it changes the cell, and it becomes an induced pluripotent cell. And in reality, that's of course a cartoon, in reality, this is what it happens. You take fibroblasts that look like that when you culture them, and then 20 days later, they become an iPS cell. And that iPS cell, you can convert it into whatever you want. And why this is important? Why this became such a, the guy that did this, and Shinya Yamanaka, won the Nobel Prize for this in 2012. So why this is so important? Because now you can take, I will give you a real case. So there was this kid that had arrhythmias. So the heart was beating irregularly. And basically, very difficult to treat because a kid. So they took skin cells, they make iPS cells from there, those pluripotent cells, they reprogram them. And then from that pluripotent cells, they make heart cells. As I said, the skin cells have the same information than the heart cells. The only difference is that they are using different information, yes? Now, if there was a mutation, the mutation is in every cell of the body. The skin is okay because it's not using that book, but the heart was wrong. So when you take a skin cell, you make an iPS cell, and then you go back and make another heart cell, now you have what we call a disease in a dish. You have a model of that specific person in your lab. So they took that, they test every possible drug for arrhythmias until they found the one that worked for that patient. And then they give the patient the right drug, and the kid got cured. And this is not, again, this is happening now. Companies, pharmaceutical companies are modeling all our diseases specifically, yes, for different people in their dish to find the exact drug. And this is how personalized medicine will come. And of course, the other aspect of, besides disease models and drug screens, is cell therapy. If we can produce every cell, you can take, for example, eventually in the future when somebody has a heart attack and lose cardiac cells, you will make cells from its own because you take them from the skin, you make more heart cells, and you put it back, and you will cure with their own spare parts, yes? So this is one of the big advantages of reprogramming. And my lab and part of the center will be working on trying to understand the different steps of this process. And as I said, we have talked about how cells develop or differentiate, mature, how we can differentiate them, reprogramming, yes. And this can be used for many things, as I said. So for example, in my lab, we have used it to generate what we call an iBlastoid, that is a model of an early embryo, so allow us now to look and study the first week of development, which is, of course, as you can imagine, almost impossible to study, and it has in a black box. And this will allow us in the future to find, we hope, solutions to infertility. Yes. And we also have made placenta cells, for example, where now we can study the placenta. By the way, many of you know, the placenta is part of the baby, not of the mother. The baby produced it and left it behind. And again, we don't know how the placenta is made because it's made, when it's made, the woman is not, the mother is not even realized that she's pregnant yet. So that period of what we call early placentation is extremely difficult to study. And this gives us now the opportunity to be able to study in a dish and try to solve the problems. And by the way, 30% of those early inceptions fail at that point. So if we could find ways to improve this by using these cells, it will be really good. And of course, this is a way to see it. So if now you see how I see all this, I see all these cells just using the information in different way. And this is what happened in disease. They start using the information in a different ways. Sometimes it's because they get an extra mutation like in cancer, or for example in aging, they just start accumulating different epigenetic changes that change the way that they use information. Yes. So the idea is, even if we get a disease, if we can correct and reprogram that disease back into a normal cell, we will be able to cure. And so proof of concept, and just as on my last slide, I want to show you some work that we have done in prostate cancer, where we identify those targets, those transcription factors that are key for making that cancer, and we just knock it down, meaning we eliminate them. And here you can see that when we use a control, just the tumor grows without a problem. When we target our knockdown, basically when we said to the cell, you cannot use anymore those important librarians, the tumor almost disappears. So in summary, I will not repeat all what I talked because Luciano has to talk and he will tell you about amazing technology that we use to identify single cells. But in summary, basic research matters, yes. Understanding these mechanisms, yes, is what allows us to progress and find solutions to new problems or problems that were there, yes. So I want to finish saying thank you to my lab. Yes, I just moved to Adelaide and set up the Adelaide Center of Epigenetics and the cancer program, epigenetic cancer program in SAiGENCI. And yes, and now our task is to build this type of research here in Adelaide, and we are very excited to do this. So thank you, and I guess you come.
Fantastic. Thank you very much, Jose. It's really fantastic to hear about what really the background of all that epigenetics work that you're going to be building. So I would now like to introduce our second speaker, Associate Professor Luciano Martelotto. Luciano is establishing the Single Cell and Spatial Technologies Lab within the ACE and SAiGENCI. Previously, Associate Professor Martelotto was the Scientific Director of the Single Cell Core at Harvard University, and he has a robust interdisciplinary scientific background, having specialized in molecular biology and biochemistry with a really strong background in technology and engineering. So please join me in welcoming Luciano.
Hello, everyone. Can you hear me well? Yeah, good. So I don't know if I can express how excited I am to be here, especially for two things. First, just to second Jose in his talk, and the other is because I think at this point in science, it's just amazing, the kind of things that we can do today in terms of science, how we can actually take things and tease them apart is something that it's science fiction from whatever angle you can see it. And today I'm going to tell a story, or at least I'm going to tell the kind of things that we do that I still think that is science fiction. Sometimes I need to kind of pinch myself to understand that this is the kind of thing that we could do. As you can see, Jose explained how he can take a cell and make from skin to make another heart cell. And that's something that it is science fiction. Okay, this is something that we would have seen potentially in something like Jurassic Park or something like that. So and today, so there are a couple of type of scientists. Jose is the kind of the guy that goes into understand every single step of the way from what a cell is and where it goes, right? But there's all the type of scientists that are actually also there, which are the kind of scientists that need to come up with these technologies to be able to help other scientists and also to their own science, to understand, to find the tools to do these kind of marvelous things that you just saw. I'm one of those, right? So in case I wasn't clear. So one thing that I want to do is before I go into a lot of details, I want to give you a little bit of brief of history about the kind of things that I do. So what I do is something that is a field called as omics. And omics come from a very, 1920s, this guy came up with this word just to sort of globalize anything that is collectively studied, all right? So for example, you mentioned, Jose was saying that any change in any epigenetic change, it's something that is a modification that happens in the DNA, all right? So when you take all of these modifications together, that's the epigenome, okay? Now, if you take all the proteins of a cell, that's the proteome. If you take all the transcript without the RNA coming from a cell, that's the transcriptome, and so on and so forth, okay? So now, in the currently, there was another work which is coming, which is the omics, which is essentially come from when a few years back, or actually, more than 10 years ago, the whole genome was, which is engaged when the DNA from our body was completely decoded or sequenced. There was this new word called genome that came up, and then the study of, or the field of study of the biology of that genome was called genomics. All right. So now what we can do is just start combining a few things. Like, for example, if you do the transcripts and we mix it with the omics, that would be transcriptomics. If we do the protein, it's going to be proteomics, and that's the study of all of the biology of the proteins, and so on and so forth. Then what we can do as well is we take the type of technologies that we use. In this case, for example, if you take cells and you take the DNA of that cells and then you study that cell, all that DNA all mixed up, that is called bulk omics. But if you take a single cell from that and you do the same thing, that's called single cell omics, okay? And it could be single cell genomics, single cell proteomics, okay? So now you need to memorize that. No, I'm just joking, you don't need to memorize this. But I wanted to say there is a lot of complexity in the type of studies that we do, and I wanted to give you a little bit of terminology so you understand what I'm saying after. So now, this is what happened, what used to happen, what is up until today, or actually a few years ago, this was the state of the art. We knew that the human has 32 or nearly 40 million, 37.2 trillion cells. That it's a lot of cells. In your body, that's the amount of cells that you have. All right? And imagine all the changes that Jose was saying that every cell can have. But you have 37.2 trillion. That it's a huge number. So now, and those cells, you have several types of cells in there. There are nearly 200 or more cells, different type of cells, skin cells, epithelial cells, endothelial cells. There is a lot of different cell types. And all of the different cell types, even though they have the same DNA, as Jose was saying, they open the books differently, all right? And that's why we have all that many types. But at the same time, now when we use this bulk omics, which now you know what it is when you take all the cells together, you mash it up and extract the information from that, and you do that at the same time, what we get is an average signal, okay? So we get all these cells, you mash it up, it doesn't matter if you put a cell of the heart or a cell of the brain or a cell of the, the liver, and you mash it up, that it's just an average, okay? But you will not know how many cells coming from the liver, how many cells coming from the heart. You will not know, all right? So, and that's what used to happen with bulk omics, okay? So we needed to come up with something different, but that's the story I'm going to tell you now. So, but the reason that happened with this individuality, that was in around 2011, actually happened a little bit before in the 90s, but everybody ignored that. But in 2011, there were a few papers that came up saying, okay, now we can do this. We can take individual cells and study those cells. But the reason why, even though we knew about these cells for many, many years, we could not do that is because there was a lack of technology. There was no sufficient technology or understanding of this to be able to tease apart these tissues and study individual cells. And this, a fellow Australian said once, progress in science depends on new techniques, new discoveries, and new ideas, and probably in that order. So this guy, Sidney Brenner, that this is in Australia, this is amazing understanding of what was important to understand science, all right? So you need to create technologies. You need to be able to come up with things that allows you or new ways to study new algorithms. Either we're studying computational biology, we're doing epigenomics, or we're doing genetics. We need to come up with new technologies to be able to create new discoveries or new ideas. And then it becomes a little bit of a sort of like inertia. New technology create new knowledge, new technology, and so on and so forth. That's what technology is so important. So what we learned so far is that biology is immensely complex. As I said, we have 40 trillion cells. And then within that cell, okay, we have DNA. And that DNA, as what Jose was showing at the beginning, that you saw there were A, C, T, G. These are the basis of the DNA. This is how the DNA is, how the information is decoded in those 4 letters, a combination of 4 letters. But we have 6 billion of those letters in one cell. 6 billion of that, all right? And each cell has its own epigenetic program, like as Jose said, some of these things are activate things, some of the things just closes the book in another cell. So that makes things even more and more complex. Then what we have is, within those cells, at any given time, we have nearly 50,000 molecules of messenger RNA. What is the messenger RNA? It's a molecule that it's a copy of the DNA that carries a message. And the message then eventually is going to be translated into another molecule, which is the protein, okay? And remember, if you take all the proteins, that becomes proteome. Well done, excellent. So these are, at any given time, we have that amount of molecules going around. That is in one cell, eh? And at the same time, all of these 50,000 molecules can convert or translate into thousands of other proteins. And if you think about the immune system, every cell in the immune system, just to make it even more complex, we have this own identity, okay? So things in the body becomes really complex. So we cannot think anymore about bulk omics. We have to start separating things out, because if we take them all together, we are losing all these details. And if we lose that detail, we won't understand anything, okay? We understand a lot of information, a lot of things. It's been very useful. But we are in an era in which we need to change things. We need to improve things. So one of the things in my lab, or one of the things that I wanted to do, is just to be able to move from the typical average, which is mash up everything together and just measuring anything that is bulk omics, to a high resolution, which means taking these things apart, okay? And this is an example. For example, you can take any tissue, any organ, or any disease model, like the one that Jose was mentioning, and then if you take a sample of that, let's imagine the lung here, and you take that and you measure up, and then you do what we do, and then we get an average signal, and that's what you will see, something that is a mix of everything, but it doesn't tell you anything about that, okay? Unless that you have one of those particular components here, maybe that it may tell you something. But what we want to get is to something like that. Don't worry about what that is, but this is the way that we see the data, all right? So it looks complicated, but what I can tell you is now, as you can see, every little sort of funny shape there, it's a group of cells that belong to the same class. So instead of having an individual, like a little dot with all of them is combined, so what we have is individual populations that we can actually isolate, like that. We can actually see them. And every little dot, I don't know if you can see, but every little dot represents one individual cell, okay? So in the case of cancer, for example, if you look at breast cancer, this is an example of a breast cancer in which you have, for many years, people would think, oh, cancer is just a crazy thing that grows and everything is the same. But in the end, what happens in reality is that, yeah, you have cancer cells, and every cancer cell, it may be different from each other, okay? At the genetic level or at the epigenetic level, but you also have other cells in there. You have normal cells that we call stroma, cells that are not necessarily cancer, but they are part of the structure, okay? Part of the entity. So you have a blood vessel, so you have what is known endothelial cells, or you may have also lymphocytes that are trying to fight the cancer, although they are recruited to do something. And you have other cells involved in all this, which are all more or less the list that is in there. So what we want to do, right? So if we take that and you measure up, we won't be able to see this complexity that we see in real life in a tumor. But what we want to be able to do is to be able to find a technology that can separate those cells into the individual types, okay? And then when we have that, what we can do is say, okay, what's the difference between those cells and the other cells, okay? We can do that computationally. So there is a lot of computational technologies around there that we can use to identify those things and separate them here and there and say, okay, well, this cell it looks normal, but it doesn't behave normal. And if we compare this one with a normal lung cell, it will probably behave different. So we start comparing things, okay? And this is about what we do. We try to compare normal versus diseased and allow us to identify what is the path between one state to the next state, okay? But to do that, we need to have that level of separation, okay? If we have all matched up, we won't be able to do that. And we are at a point in technology where we can do that. So another way that I always like to explain how the technology that we use is, which I haven't even mentioned that yet, but I will tell you what it is that imagine that you have a complex tissue like that. And now imagine that complex tissue like a ball of fruit, okay? Where every fruit represents a cell or a part of that tissue, okay? Now, what we can do with that is if we do bulk omics, we can make smoothies, all right? So we take that, make little preparations there, and we taste it. Some of you may say, I can taste orange, maybe kiwi, banana, but then as you keep putting more and more, you're gonna lose this ability to know about exactly what was in there. You can more or less infer, but you will not know for sure. You will not know if you have 3 bananas or if you have 1 banana or if you have 3 kiwis, okay? So that's what the comparison with bulk omics does. It gives you an average. Now, there are other technologies that allows you to separate the bananas from the pear, from the apples, all right? That is what in our field is called cytometry. It measures a cell, it measures a property of the cell and separates that. So now we're gonna have different smoothie. We have the smoothie of kiwi, the berries, and so on and so forth. But still, we don't know what's going on within each one of those smoothies. We know that how many of those smoothies we have, but we don't know how many of those apples or bananas were in those individual smoothies. But then we have the technology that I love the most, which is single-cell omics, and which allows you to take all the little pieces and separate it into individual entities. And now we are really getting what we call granularity. We can see the individual cells and how different they are from each other, okay? But then we can do something even cooler than that. Because at the moment, what I'm doing is taking the ball, breaking it apart, okay? And that's what happened in the tissue. I take the tissue, take it from a patient, and then separate it into individual cells, and then I study those cells, okay? But those cells lost something that is very important, which is the original position in the tissue, all right? And the cells are not like floating around. They are in a structure. They are in a position for a reason, okay? They are not on the left side. They are on the right side for a reason. It's up here or next to the vessel for a reason. Because when people started thinking about that, I said, we need to do something else. We need to repeat what we're doing with single-cell omics, but we need to put it at the level of the space. And that's when the spatial omics came up, which is essentially like looking at all of that in a chart. And you can see that you have a certain order of all of these things around, okay? And this is the position of those things is not random. Someone chose to put those things. And in a tissue, it's our own body who chooses where to go, and there is cell signaling that happens between cells, and that you should go there or you should go there. But that positioning, it's very, very important, okay? So, and the two technologies that we are capable to do here I would say, to the fullest is spatial omics and single-cell omics. And here I'm saying we can do every single one of these metabolites that we were mentioning in the two states, as a single cell and also in space. And what that will allow us to do is we will be able to characterize different cell populations that before we didn't know existed, and even if we knew that existed, we can characterize them more accurately and also in space, okay, within the original tissue. We don't have to stress the cells when we take them out. We have it right in there. We will be able to see what cells are interacting with each other, okay? In which, it's not going to be the same if a cell is close to a vessel, to be far away from the vessel, to be close to a normal cell, to be far away from a normal cell. So we are going to see all this regulatory signaling between cells and also within the cells, okay? We will be able to see different cell types that we probably never discovered before because they were hidden, because they were probably very rare. And if they are rare and you mix it all together, you won't see them. But if you are able to separate them, you will be able to see them. Now, cell state is also another thing that I haven't touched on, but cell state is, you can have a cell in a type of cell, but that cell may be moved in what we call the transcriptional space, which means if you have more RNA expressed for whatever reason, it's not the same that before you actually express. It's the same cell, but it's in a different state. So that creates more complexity, what I was mentioning before. And also, we may discover new cell types, okay? And the other thing that we can do is we can say, okay, well, these are all the cells that we have here. How all these cells relate to each other? Because that is important to know. If we have a particular cancer, all right, and we look at a particular cell, and that cell, we want to know who or what cell gave rise to that cell, to that group of cells that actually probably are resistant to therapy. So we want to know where they come from, because if we know where they come from, we can understand them, or potentially we can target them, okay? And then the other thing that we can do is we can profile, and with single cell, so profile meaning we can understand that to a lot of detail, healthy and diseased, and organs and tissues. And this is part of what is known as the Human Cell Atlas, all right, in which we have been, is a global, like in the whole world, effort to take every single piece of every tissue and study every single cell in that tissue and characterize each one of those, and also characterize the interaction amongst them, and also their position in space. So, and that's why I think that this is a very powerful technology, these powerful technologies that will allow, we can put them all these technologies together to start paving the road to find finally a cure, or at least to approximate the cure, and what I like to call one cell at a time, and wherever they were in space. And with that, I hope you enjoyed it, and thank you very much, everyone.
Thank you very much, Luciano. We now do have some time for our Q&A session, so I'd like to invite Jose back up here. So that was fantastic, both of you. It's really exciting to hear about this really, really cutting-edge knowledge and technology that you've brought to Adelaide. So I think we're going to start off with some pre-submitted questions, but we will also have a couple of microphones roving around. I will just also like to make a quick comment, which was with the pre-submitted questions, we did receive a few that were personal and about people's own personal experience with cancer. So we decided that we would, rather than include those, we'd prefer to keep the questions quite broad and general, and really keep them quite overarching so that they can benefit everybody. But I thought maybe before we get into these pre-submitted questions, you've both done a really fantastic job of speaking about the technologies and the epigenetics and the new programs. But I wonder if I could ask each of you to make a few comments about how your work's going to sit within SAiGENCI, and maybe how you're going to employ the epigenetics research and the spatial technologies in the study of cancer specifically. If I can ask you to just a few words about that.
So, as we try to explain, cancers, they change the normal program of a cell. So a cell becomes cancerous when the normal program fails. And usually it's because they have mutations, so it's basically you grab a part of the book, and it's gone, and now the cell, basically that gene, that book, does a function that they shouldn't be doing, or literally two books get smashed half and half, I don't know, and you end up with little red hood going into I don't know, Alice in Wonderland, and the wolf make a disaster there, and things like that. So, but that, regardless what it does, that will change the epigenome of the cell. So rather, for the last 30 years or 40 years, people have focused mostly on those mutations. And our approach is to see it from, okay, let's forget for a second of the mutation as a primary target, and let's see what is changing in that epigenome. Because if we can control the epigenome, we are 100% sure, like we have evidence, yes, that we can control the cell state, as I show you. Yes, if we can make a skin cell become a neuron, a cell of the brain, we can definitely make a cancer cell stop being a cancer cell or becoming a neuron. Like, why not? So, and so that's how we are going to employ. So our way to, in SAiGENCI, yes, our work in cancer will be to use that epigenetic view, which is not only mine. Yes, there is a whole field of cancer epigenetics that looks at this problem from that perspective, and that's what we are going to do. We are actually recruiting other group leaders to work in epigenetic and cancer, yes, and try to build from there that perspective here in Adelaide.
That's fantastic. It's really exciting.
Well, what can I say to that, right? But essentially, yes, what he said. Now, I mean, the idea of us is just to build a community that, you know, within SAiGENCI and ACE and the University of Adelaide, in which, I mean, South Australia in general, and we can collaborate and we can educate and we can create the, you know, future leaders that can continue and work with us and continue our work whenever we are not here anymore, which I hope is not soon.
Well, there's been a pre-submitted question that leads into that really well, which is from Artemi, and they ask, what student involvement is there going to be in SAiGENCI research projects? Are you recruiting? That's probably what they were hoping.
Yes, we are hoping to recruit good students, and those parts we are going to make them good. So yes, we are actively recruiting at the moment, you know, PhD students, honor students. So these are the things that we are aiming, and yeah.
Actually, yes, we have, and SAiGENCI will have, like, of course, we are part of the Faculty of Medicine, so that's our PhD program, yes, but it will have their own cohort of students that will be focusing their research as a one thing into cancer, and of course we have our own idea how that program, or sub-program, let's call it, will be run and evolve inside the university.
Yeah, fantastic. So we do have, I'll open it up to questions from the audience now. Is there anyone who's got any questions at this stage? Don't be afraid to pop your hand up. We've got one in the middle.
Hi, my name's Richard. I work in the Faculty of Health here. Question for Jose. I was just wondering if you can change the landscape to turn, for example, a skin cell into a neuron, can you change the landscape to turn a cell into a better version of itself? So could you take a muscle cell and turn it into a stronger muscle cell, or a disease-resistant cell, or can you only create cells that already exist in the body?
Yes, that's an excellent question, actually. So, well, in theory, yes, and there is a whole, yes, so I will answer that in different steps. So, for example, we have work in grabbing, getting a cell of the intestine that is aged, yes, and therefore doesn't make your intestine gets, every 4 days gets completely replenished. So that's how the part of the intestine gets. And when people age, that function starts to diminish and diminish and diminish. And what we have found is a way to reprogram that into giving the same function of a young cell. So now it can rebuild. So it's more or less what you are saying. And actually, when we apply that to a young cell, it also replenish itself faster. So we give them like we enhance it, the capacity to the potential to differentiate and make more of these cells. So that's one. Of course, there is something, I'm not a muscle biologist, yes, so I know that, for example, some things that can get you muscle strength are mutations in the myosin chain, yes. But yes, I'm sure you can make, for example, bone strengths. And we have tried to get funding to do this, for example, to get bone strength, to basically increase the amount, changing so inside your bones you have two types of cells, osteoclast and osteoblast, and they are constantly rebuilding your bone and degrading your bone. And our hypothesis is that if you change this balance, you can make the bone stronger or weaker. Yes. So yes, you could reprogram to get a stronger bone, for example. Yes. And then there is the field continue. If you keep going, there is this field becomes synthetic biology, where you can say, okay, why we don't have a cell that has these two different functions? Yes, and this is a cell that doesn't exist actually, but maybe for some medical applications, you don't want the cell that exists. So for example, you could say, if you have diabetes, you need a cell that makes insulin, yes? And maybe you also want that cell to make, I don't know, another hormone or something else to control other disease. And you make a cell basically that makes, I don't know, thyroid hormone and insulin, which will be a mix of thyroid and pancreatic beta cells, in one cell, because that's what you need. So yes, there is no reason why you cannot activate different functions and get these cells. And that's what a lot of the field is trying to go to. So really nice question.
Yeah, it's a really exciting area.
Yeah.
Are there any other questions from the audience?
Yeah, thank you both for your informative presentations. This one will be for Professor Polo. You put up a diagram showing a pluripotent cell being differentiated into a cardiac cell and uses a disease model. The presumption there is that differentiated cell would be in a disease state, or is that always necessarily the case?
No. So yes, it's a good question as well. So if the disease is a genetic disease, and it's what we call cell autonomous, meaning you get that gene mutated, and the cell, that cell, no matter what, will have the disease, yes, it will happen that. So, for example, many of these arrhythmias are mutations that you are born in the whole body with mutations in some of these ion channels or receptors that generate a cell autonomous problem in the beating, yes, or in the cell. So that will be. But some, as we know, many of our diseases are environmental control. So, but in that case, you can also model it, because what you can do is emulate the environmental conditions. So, for example, if some disease arises because of high glucose, well, you can put more glucose, and that is a sugar, you can put more sugar in that place, yes, and you model. And, or for example, UV irradiation from the sun, of course, you can create skin cell and irradiate it with UV, yeah, this is done a lot, and see how that UV modify that. So there is a mix. If it's genetic, you will represent it. Otherwise, if it's environmental, you can put it there as well. Yeah.
Good, thank you.
Okay.
We'll come down to the front here.
Question for Luciano. As an engineer, I was taken by your description of the process of new procedures lead to new discoveries, lead to new ideas. Could you just briefly describe some of the technology you're using and how you see it developing in future and where it's come from to allow you to make these new discoveries, particularly at such high levels of resolution?
Yeah, so for example, I can tell you how the single cell gave rise to this patient, okay? So essentially, without going into the detail of how that works, so essentially what we used to do originally was to take the tissue, dissociate it. What we were thinking is how to separate those cells, all right? So what keep the cells together, someone discovered that what keep the cells together was something called extracellular matrix, okay? So, and now with that knowledge, then we were able to find out, okay, so what is this extracellular matrix made of? And it's made of a lot of proteins and some sugars. So what we started doing is just to combine different enzymes that we knew would target those particular polymers that are part of that structure, and then we started separating the cells. So by discovering what the extracellular matrix was, we were able to use some tools that were available to dissociate these cells. Okay, so that's one way of seeing this. Then we started separating those cells and analyzing how we can now study, for example, how the genes are activated and deactivated, like what Jose was saying. Okay, so for that we started saying, okay, is there any way that we can take what we already know from taking the bulk omics, right? We measure apart and take all of that. Can we do that in single cell level? So what we needed to do is we went into the field of microfluidics, which you're probably familiar as an engineer. So the one way to do that is instead putting cells one by one into individual things, what we were doing is we were running the cells through different tiny little channels in which only one cell will fit. And then all cells will align one after the other, and they were just going one after the other, all right? But then we needed to do something else. We needed to create a chamber where the reaction happened, like the chemical reaction happened. So we figured it out, well, not me, but someone figured out, but I feel part of the population. So what they figured out is that if you can run another channel that has all the reagents that are required for that, at some point they can connect the two channels in some point, right? But then you need to create the channel, so that will mix the two things, but they won't do anything. And then someone said, oh, hang on, if we mix oil with water and we shake it, we make droplets. How about we do the same thing? We do this one channel, another channel, and oil this way, and then at some point the three come together and then generate some droplets, all right? And inside the droplet, you have a cell, you have all the reagents to do that, okay? And that becomes a microchamber. In that way, we started making that single cell technology that we wanted to do, okay? And that is how the process created new technology generating another technology. So in this case, we started from a tissue, extracellular matrix, enzymes, microfluidics, microchambers, and then from then on, we can continue until today when we can do that thousands of times better, okay? Does that make sense?
Very sci-fi. Thank you. All right, we do still have a little bit of time for maybe one or two more questions, if there's any more from the audience.
It has always been easier to cure a cell than an organism. How are you proposing to use the effects of treatment of a single cell for mostly treatment of a patient or mostly for prevention, like in vitro fertilization and so on?
Yes, please. Yes.
Or I think I heard you all right. So I think what the question was, you're talking about all these different things that are happening on a single cell level.
Yep.
How do you see that transitioning from being able to cure a single cell to being able to cure something that's at a person level?
Yes. So yeah, of course, there is two. I think that there is two questions in the question. One is, if all these are different cells, this is what I understood. If all these are different cells, yes, and you are going to cure one by one, how you go from there to the organ, yes? Well, the organ is made of many different types of cells, but they have many common things. Yeah, so maybe an organ will have, depending with which resolution you look, but it will have, let's say, 10 types of cells or 15. So, and usually in the case of cancer, there is two things that could happen. One is some of the cells, mutate and go bad, and you have one population of cells feeding this tumor, yes, like a cancer stem cell, feeding this tumor constantly. So if you find that cell, you just can target that cell type, because the rest will die anyway. Is this one the one that you need to cancer? So that's one way to answer the question. The other way to answer the question is the delivery part. Yes, one thing is to do it in the plate. I completely recognize that. The other thing is to make that a therapy. And of course, that's where a lot of also research are. So a lot of investment should be done in this initial step, finding the possible cure. Yes, so now, or the mechanism, so now, okay, now we know how this happened. And then, of course, there is what many people call the valley of death, that is, okay, now we know that we can do it in theory. How we convert that works so well in the lab, how we go all the way to a patient. And that takes years, clinical trials, and it will be great to accelerate that, but it takes a lot of time. So yes, I completely agree. There is a lot of work to do even once you find the solution. Yeah.
It's an important question to ask. All right, if there are no additional burning questions from the audience.
I have a online question.
Of course, go ahead.
From Lisa. So she's asked, do you have a time frame where you expect that your research will start to change treatment options for cancer patients?
A time frame?
Yeah, is there a time frame on when you think your research will start to change the options for cancer patients?
So, okay, depends how you answer that question. So one is from a very high view. It started already to change, yes, because disease modeling is used for cancer research. A lot of new drugs are coming in the epigenetic field that go against to find, basically there are drugs that target the epigenome, yes, and they are used nowadays, yes, for many different types of cancer, and some of those cancers are being cured with those types of drugs. So from that perspective, it's happening. Now, you are talking about my specific research. Putting a time frame will be very difficult, exactly for what the lady sitting there asked, is even when we found the mechanism, it takes a time. Nevertheless, because we work in this type of predictive biology, we have the capacity also sometimes to predict which drug that already exists can accomplish the process that we want. So what we do is we go to these databases that have these drugs that have been already approved for many other things, and of course we try first those when we try to predict computationally which of these drugs will work. So if that happens, this accelerates the process. But yes, putting a time on that, I would say hopefully as early as possible. Yes.
I just want to comment on that as well. So to be honest, it's, as Jose said, it's working right now, and I will give you an example in a second. But what is important to probably know that is these things usually take time to be accepted, mostly to be applicable to patients. Okay, so but things are happening. So for example, when I was doing my postdoc, this guy came to me with an idea, which is, I think it was lung cancer, and this particular lung cancer was driven by a particular change in the DNA that make the cells divide very, very, very fast. Okay, which is usually one of the hallmarks of cancer, just the uncontrolled proliferation. But what was particular about this change is that what the cells were doing is acquiring many copies of that particular gene. All right, so that change wasn't any change; it was increasing the number of versions of that gene. So, which means that that was one way of the cells to make that gene activated. So, one thing is just to express a lot of RNA from that just by activating the gene, like opening the book, like Jose was saying. Another way is to get in the same book, but many, many copies of that thing. Many, many, many copies of the same book in one space, okay? And that is called gene amplification. It's a gene that becomes many, many times copied. So what we found using single cell genomics at the time, and I'm talking about 2017, was that depending how you treat depending how you give the drugs to the patient, that population of cells that having many copies of that, you can control. So you can control those cells that have many copies of that. And by controlling that, you keep that on check, and then you can add another drug that is stronger, and you can kill the whole thing. Okay? Obviously, that was in the lab. Okay? It wasn't something that is used in patients, but that changed the way that now oncologists are looking at how to treat patients. Instead of obliterating the entire population, what they do is they give serial doses. It was actually an old school way of doing this, but for this particular cancer, for this particular mutation, it seems to work. So they do a combination of drugs in which you do not kill the whole thing. You keep those cells that have the ability to replicate and create many copies in check because they are competing with another population, and then you just target them, all right? So, but that can only be seen when you do use these technologies, all right? So previously to that, I was like, we just try to kill everything. But what we try to do is just to find ways so the patient doesn't get too much therapy if it is not needed, okay?
I think that's a really fantastic example of how that work. It really is affecting the way we treat patients now. We are unfortunately out of time tonight, so I would like you all to please join me in thanking our fantastic speakers, Jose and Luciano. And thank you to you, our audience, both online and in person. Thank you for your fantastic questions and comments tonight. We look forward to inviting you back in May, when we're going to hear from 2 researchers looking into dryland agriculture for space and extreme environments, which sounds really cool. So be sure to sign up for our mailing list so you can receive the latest information about all of that and other Research Tuesdays news. Thank you all very much for tuning in once again, and we hope to see you next time. Good night.